Alright, so the second step of aerobic cellular respiration is going to be the Krebs cycle, also called the citric acid cycle. Now recall from the previous glycolysis video, we took our sugar, we converted it into 2 pyruvate molecules, and now we are going to take those and they actually have to enter the mitochondrion to be further oxidized. So what we're going to do here is bring them into the interior of the mitochondrion called the matrix. Okay? So, even though you will not see oxygen directly used in the Krebs cycle, this does require oxygen indirectly. So keep that in mind. Okay. So here we go. This is going to be the outer membrane of our mitochondrion, and then here we have the convoluted inner membrane. Okay, so here comes your pyruvate. We're going to do this one pyruvate at a time. Just realize that the same thing is going to happen to it again. Alright, the pyruvate diffuses across both membranes, outer membrane, inner membrane. Pretty much immediately upon entering the matrix, a carbon is going to be removed in the form of carbon dioxide. Don't worry about where the oxygen comes from, the carbon is most important there. This carbon dioxide is a waste product of the reaction. Recall that glucose in the presence of oxygen will yield carbon dioxide, water, and ATP. This is the carbon dioxide that is produced as a waste product right there. It's going to come from the Krebs cycle. Okay, that is going to produce a 2-carbon molecule, which is then going to be oxidized, that is, NAD plus is going to remove electrons from that molecule, producing NADH. Okay, this 2-carbon fragment is then going to be attached to a molecule that we call coenzyme A, and collectively this molecule is called acetyl-CoA. This acetyl-CoA is then going to take this 2-carbon fragment and attach it to a 4-carbon fragment that's already sort of present in the matrix. So when this molecule takes these 2 carbons and attaches it to these 4 carbons, it's going to result in a 6-carbon molecule, and this is citric acid. Okay. When the 2-carbon attaches to the 4-carbon, this acetyl-CoA will actually return and pick up a new 2-carbon fragment and perform that same attachment. So it keeps cycling. It takes a 2, puts it to the 4, makes citric acid, returns and grabs another 2-carbon, and just keeps repeating. Okay, but now we have our citric acid produced, and from here we're going to perform a series of oxidation steps, as well as remove some carbon for carbon dioxide and produce a little bit of ATP by substrate level phosphorylation. So here we go. Here's how that's going to look. We're going to take, first thing, remove a carbon in the form of carbon dioxide. That's going to result in a 5-carbon molecule. We are then going to have a redox reaction where NAD plus removes electrons to create NADH. We are then going to remove another carbon in the form of carbon dioxide. We need to have another oxidation event where we produce another molecule of NADH from NAD+. We get the formation of ATP, and again that's going to be substrate level phosphorylation. Essentially we have the transfer of an inorganic phosphate to ADP, and that's going to be by an enzyme transferring it from one substrate to the ADP. Here we go with another oxidation event, producing more NADH from NAD+. And again, what we're doing is pulling those electrons off that molecule. That's called oxidation. And then, last but not least, we have another oxidation event where the molecule FAD pulls electrons to create FADH2. Okay, so it seems like a fairly complicated cycle, but let's just go over some of the, let's kind of summarize this. What are the main things we get out of this? Okay, we get the production of carbon dioxide. 1, 2, 3. Now that's going to be 3 carbon dioxides from one pyruvate, so you can obviously double that to 6 if you want, did the other pyruvate. And then we get the formation of our high energy electron carriers, NADH. Recall we got one in the beginning when we first started, NADH. We get the production of an NADH molecule here. We have the production of NADH here. And we have the production of NADH here. Okay? That's 4 NADH molecules. 2 pyruvates would give you 8 total. Just double it. Okay? Then we get the production of FADH2 right here, which more or less functions the same way as the NADH. It's going to carry those high energy electrons to the next step as well. And we also get the formation of ATP. Okay, it's going to be 1 ATP per pyruvate, so if we send the other pyruvate into this, we're going to get a total of 2 ATP by substrate level phosphorylation. So again, per glucose, 6 carbon dioxide, 8 NADH, 2 FADH2, and 2 ATP.